Hello, everybody. I am going to be talking about um, the sensory systems. So, we'll talk about the nose and we will talk about the tongue and hearing and eyes. That's what we'll talk about. Okay, start with the nose. Let me draw a little thing here. Best nose you've ever seen in your whole life. Okay, so here is the nasal cavity, right behind the nose. In the nasal cavity, you have olfactory nerves that enter the nasal cavity. At the top of right above the nasal cavity, in between the I don't know, I'm gonna butcher the pronunciation here, but the cribriform plate, and the cribriform plate separates the olfactory bulb and the nasal cavity. Make sure that's working. Okay, that is the nasal cavity, the nerves, and the olfactory bulb. And then the olfactory epithelium. Let me get that. The olfactory epithelium is it's kind of like this space right here where all of this connects. It is it's where you have the inside the olfactory epithelium, you have the cribriform plate, you have the axons with the olfactory, um, with the axons with the olfactory nerves, and then above it you have the olfactory bulb and the olfactory tract that leads to all of that, and leads to the nerves lead to everything. The with this is like this is like anatomy, but with the as we go more into physiology, when we smell things, we have chemoreception that detects chemicals. So inside of the nose, we have odor molecules that will dissolve into the mucus layer, and they'll bind to receptors on the olfactory receptor neurons inside of this epithelium, olfactory epithelium. So here in the nasal cavity. So we have odor molecules will enter, go to this receptor, which is linked to the neuron. Okay. Each receptor is a G protein coupled receptor. When an odor binds, the neuron will depolarize and send an action potential through the cribriform plate into the olfactory bulb. The bulb will then recognize the signals, and that's how the brain interprets different smells. Now, on the same with using the same thing, chemoreceptors, we're also going to describe the tongue. So with the tongue, we can have sweet, salty, sour, bitter, and umami. Those are the those are the taste regions, or the aspects of taste. Taste is also a chemical detection. So the molecules in food will dissolve in saliva and enter taste pores on these taste buds. There are different receptor mechanisms. So sweet, bitter, and umami use G protein pathways, while salty and sour use ion channels. And when these cells depolarize, they release neurotransmitters onto sensory neurons, and that produces action potentials to the brain. Okay. Now, we're going to turn a little bit to hearing. So we'll have sound waves enter the ear canal. When the sound hits the tympanic membrane, it vibrates. We have three ossicles, which is malleus, incus, and stapes. The malleus, incus, and stapes act as a lever system that amplify these vibrations. The stapes pushes the oval window into the cochlea. In the cochlea, it's a spiral. This wave that the stapes is called the paralymph wave, and when the oval window moves, it creates pressure waves in the fluid of the cochlea. Okay, and then inside the ear, you also have hair follicles. These are hair cells that have sterocilia. When fluid moves from the basal membrane, when the fluid waves from the cochlea move into the basilar membrane, their hair cells will sit on this membrane, and their sterocilia will bend toward the tectorial membrane. Okay, this bending there's different sizes of these hair follicles. The highest one will cause these cells to bend. So higher pitched sounds will cause cells to bend. As they bend, potassium rushes in and depolarizes these hair cells. The cell will then release glutamate into the cochlear nerve fibers, which then fire action potentials into the brain. Okay. Now we're going to go into the eye. So the eye has a few different parts to it. We have the lens, we have the cornea, and the retina. The eye focuses light using two structures, the cornea and the lens. So light will enter, and the cornea does most of the bending, and the lens fine-tune the focus. When the ciliary muscle contracts, the suspensory ligaments around the eye loosen, and the loose ligament lets the lens become thicker and focus on near objects. And so this is, you have a very thick lens. When it relaxes, when the ciliary muscles relax, the ligaments pull tight, and then making the lens flatter. That's the ligament. So pulling those ligaments tight, making the lens flatter, and this focuses on far distances. Okay. Now inside of the eye, you have rods and you have cones. Rods work well in low light, and will detect shades of gray, whereas cones need bright light and detect color like red, green, blue, that kind of stuff. Okay. So, in darkness, rods and cones are they're depolarized and continuously release glutamate. When the light hits their photopigments, the retinal molecule changes shape. This activates the G protein cascade, which closes sodium-potassium channels and hyperpolarizes cells. This decreases glutamate release signals in bipolar cells, which then excite ganglion cells. Ganglion cells will produce action potentials that travel through the optic nerve. With these rods and cones, we'll have center and surround zones. Rods, cones, or no. Anyway, the inside the center, you can have an on center of the cell. If it's an on center of the cell, the light in the center increases activity, while light in the surround decreases activity. And then off center works the opposite way. This setup, it enhances contrast and helps us detect edge and shapes in our visual field. Okay, so I think that pretty much wraps things up for today. Thanks for listening. Oh, that's my stuff.